Welcome to this interview with Shane Needham, who is actually the co-founder, president, and CEO of Veloxity Labs, who is based in Peoria, Illinois. And today we will talk about bioanalysis. Welcome to be with us, Shane, today.
Thank you. Thank you for having me, Nando.
So it's our pleasure. So then thank you for taking the time to speak with us today. We are really interested in understanding how your team approaches bioanalysis workflows and where you see opportunities for improvement. High-resolution mass spectrometry has been evolving quickly, and we would like to explore how it's impacting both the science and the business side of your work. We'll start with some general questions about workflows, then move into specific areas like selectivity, signal-to-noise, multidimensional separation for peptides, and applications with GLP-1s. Our goal is to connect the technical advances with the commercial outcomes that matter most to you. So why we are here? Because it's very important to say that Veloxity Labs has been the first CRO so purchasing the 86, so the new technology for high resolution from SciEx. And yeah, so then congratulations. So
thank you.
And we want to see how it performs in your hands, Shane. Thank you very much again to be with us.
Thank you, and it's a great collaboration with SciEx and your team. Thanks again.
Thank you. So first question, in your current bioanalysis workflows, what challenges do you face, and how do you see high-resolution mass spectrometry technology helping to address them?
Well, I think from the beginning, especially with a CRO and getting therapeutics to market for biopharma and pharmaceutical organizations, it's all about speed. And Veloxity is all about speed. Velox means speed in Latin, and so Veloxity is all about speed. The faster we can get therapeutics to market, the quicker we can treat disease. And that's our vision at Veloxity, is treat disease one sample at a time. So it's all about speed. So why does, for example, mass spectrometry, especially high resolution accurate mass spectrometry, work better? Why? Because we can now get more selectivity that gets us more signal to noise. Some people correlate that to sensitivity. Therefore, we can do faster method development, better methods and faster methods, and be faster overall. So it's all about speed. So sensitivity is a byproduct of now we can get more speed.
Mm-hmm. You talk already about selectivity and signal to noise, right? To get a better signal to noise. So then how those two factors impact into the reliability and efficiency of your bioanalytical results?
So when you think about signal to noise, analytical chemistry throughout time has always been about signal to noise. Can you detect something that's greater than the noise level, right? And so it's always been about that. And believe me, I have had throughout my 30 or 35 years in analytical chemistry, we have vendors always asking, do you need more sensitivity? Is it enough? No, never. We will always need more sensitivity because we always need more signal to noise. Now, with a HRAM, so high resolution accurate mass, what that does, it gives us better selectivity. So we can differentiate more masses from each other to differentiate them from the noise, so that in theory gets us better signal to noise. It is another tool to improve selectivity without, for example, more laborious extractions or more laborious longer chromatography. We can do it automatically by using HR, high resolution accurate mass.
Mm-hmm. Thank you, Shane. So we have been talking before to start the interview, so and many times even, so then about how proud are you about your team, right? So then how good is your team, right? So then especially in what concerns to the analysis, so then of peptides and new molecules that you mentioned before as well. So then one of the techniques that you are using is multidimensional separation. So then how does this multidimensional separation with instruments like the 86 improve peptide analysis workflows in your lab?
So when we think about, for example, with peptides, they're becoming much more common therapeutics because they have some of the advantages of small molecules with easier dosing, lifetime, so on and so forth, but not as difficult to scale or to commercialize or, for example, to dose as a large molecule like a monoclonal antibody. So peptides are like an in-between, right? Small molecule, large molecule, right? 4 to 5, 6,000 Daltons, something like that. So obviously, they're challenging, right? They have multiple, there's radionic, so they have acidic and basic groups, right? So in the past, you've always had to use either orthogonal type of separation or chromatography and or sample preparation. Like for example, you'd use solid phase extraction offline and then use some kind of maybe an ion exchange or something offline, solid phase extraction. And then your chromatography would be reverse phase or some type of reverse phase chromatography, right? So that's orthogonal. And that gets you better selectivity, okay? And so obviously that requires a lot more labor and a lot more intensive resources. Now that we, for example, with a high resolution accurate mass like an 8600, we can, for example, skip some of those steps or minimize the chromatography and minimize the orthogonal sample preparation to possibly, for many projects, maybe just do a protein crash. So a simple type of sample preparation that they can go right into the high selectivity of the 8600 high resolution accurate mass instrument. And so it really streamlines and minimizes necessarily the need for orthogonal or multidimensional separations. It doesn't say it's going to take it away. Chromatography and sample preparation will always be important. So this is just an 8600, like with high resolution accurate mass, is another tool to optimize the selectivity.
Mm-hmm. Talking about, and you talk about selectivity, you talked before about time, right? And as well. So then, and automation usually. So then it helps in some way to free up the staff time, right? So then, and the question, it's related to this. And which parts of the workflow are most automated now, and how does that free up the staff time?
Well, I think that's a great question, Nando. And so now that we don't have to spend as much time in the labor of sample preparation and even method development and chromatography, method development especially is very difficult to automate because it requires a lot of thinking, of changing columns, of changing mobile phases, checking data, so on and so forth, right? And orthogonal or offline sample preparation, it can be automated, but again, it's more difficult than what we know can easily be automated is LC-MS. We put our samples in a rack with an auto sampler, and they go right into an instrument like the 8600 high resolution accurate mass spectrometer, right? That's easily automatable. So now we've freed up all this method development time because of the extra selectivity of an 8600, and maybe minimize for sure these extra sample preparation steps or orthogonal chromatography or multidimensional chromatography. So what we've done now, because of this extra selectivity, which leads to better signal to noise, better sensitivity, now we can use our automated tools, which is LC-MS, already automated with an auto sampler, to our distinct advantage.
Nice. So, and so we talked before about peptides, but how do you see these methodologies then with the high resolution mass spectrometry and all the workforce improvements that you and your team are doing today? So then supporting the analysis of complex and very famous molecules, I would say, like GLP-1s today. So then both from the scientific and the commercial point of view.
Yeah, from the scientific and commercial point of view. So let's talk about the commercial point of view first. Okay, commercially, our bread and butter are still going to be in the quantitative biological lab like we are at Veloxity, right? They're still going to be, for example, 6,500 plus triple quad instruments, those type triple quads. And we have others in the SciEx platform as well of triple quads. But that's our bread and butter. For the majority of small molecules and things that may not be as complex, I don't see that ever changing with triple quads, right? But when we have a method development issue, or especially as you get to larger molecules like peptides and then even proteins where you may have to digest them, then that's where the 8600 comes in, because it's going to give you more selectivity because of the accurate mass. And when I say more selectivity, what I like to use is it's just different. It's different selectivity. It might, the more, what does more mean? No, it's just different. And that's what selectivity is all about. It's different than the triple quad because that's nominal mass instrument. Now we go to an accurate mass instrument, and it is going to give us different selectivity that we can differentiate certain masses that may overlap and, let's see, interfere with a nominal mass instrument. Those instruments, no nominal mass instruments, aren't going away. They're still going to be our bread and butter. But for that different selectivity that we need for peptides and proteins, it's going to be like an 8600. And so here's what we're really excited about the 8600, because we know the other SciEx platforms, 7600 and whatnot. We're really excited about EAD, for example. And I won't get into the details of, I'll let the scientists talk about that. We're talking about the science now. But the EAD, electron activated dissociation, so instead of CID, it gives us, again, a different type of fragmentation. Again, different selectivity. That can also improve our solenoids, especially with complex molecules like peptides. Now, especially with the, I don't remember the terms that SciEx is using, but the collection of the ions and then pulsing them, that's increased the sensitivity even more. And this is where we're seeing that, like I said, the 8600 commercially is really competing against those low nanogram per mil level LBA immunotype assays with mass spectrometry. That really excites me because the data is just so much more solid, so much more accurate, so much more precise and predictable with LCMS. So we can get those therapeutics to market much more quickly. So that's on the commercial side. Scientifically, peptides are challenging. 4, 5, 6,000 Daltons, maybe more. Large molecules, even more challenging if you have to digest them to make surrogate peptides, right? It's the bottom-up digestion. Again, and these are things that we're trying to figure out in our workflows, because this is large molecule bioanalysis via LCMS, especially high-resolution accurate mass, in a CRO, in a bioanalytical world with regulated workflows, is still in its infancy. So we're still trying to figure this out and how it works in our workflow. However, for example, with a mass range of 60,000 Daltons, do we not even have to digest some of these proteins now? Can we maybe not digest them and use surrogate peptides and use the original intact molecule and do true bioanalytical quantitative analysis? Hey, the future shall see. And it's certainly something we're looking at already.
Yeah.
And so it's just super exciting where we're at with this convergence of technology and the evolution of high resolution accurate mass spectrometry and mass spectrometry in general. It's great to have been a part of this process, even since using serial number 1 SciEx in the industry when I was at Pfizer. To see all the developments has been truly incredible. I'm grateful to be a part of it.
And you are, because you started with the APA 3. Right, so then you're moving into the latest one, from the first to the last.
Yeah.
And talking about the last, we are in front of facing the last question. So, from a business perspective, how do these improvements, everything that we have been talking till now, so then faster workflows, better selectivity, signal to noise, scalable peptides, and GLP-1 analysis, translate into value for your organization, so value for Veloxity and for your clients?
Well, I'm so fortunate to be surrounded by a great team at Veloxity, and what's happening already with the 8600 news, but we've already happening over the last year or so, is we're becoming the peptide experts in bioanalysis as a CRO, and so that's becoming a major source of our client concentration, because it's challenging, right? And we've shown that we can deliver. And so the 8600, with that market segment right there of GLP-1 agonists and other peptides, can be used in other therapeutic areas as well, right? And they're becoming more and more popular. It's only going to improve our customer concentration and diversity all at the same time. So we can do more projects more quickly. And super, super excited. Grateful to SciEx that they've been a great collaborator to Veloxity and me over the years. So thank you very, very much.
Thank you very much, Shane. So thank you to you. Thank you for trusting in SciEx. So thank you for the science that you are doing. Thank you for being an entrepreneur. So then, and starting new companies on and on and overall. So then, congratulations for this. So, and well, let's see. So then we will do another interview.
I
Okay.
love that idea. Excellent.
Thank you very much. Have a very
Thank
nice
you.
day. Bye now.
Thank you.